Hey guys, so today we are going to try to understand chromosome mutations and what are the various types and how does it function. So, what is a chromosome mutation in the first place? So, basically speaking, chromosome mutation stands for variations in the number of chromosomes or the structure of chromosomes. Now, before I begin, I want you to understand the basics of the structure. There are arms, right? So if I'm representing the segments of the chromosome, so this dot here, A, B, C, D, E, F, G, these letters basically represent the segment of the chromosome, whereas the dot here represents a centromere, the first constriction you can say, where the two where the chromosomes, where the DNA is very close to each other. Now, what are the basic three types of chromosomal mutations? So we summarize them as rearrangements, where the segments of the chromosome rearrange. The other one is called aneuploidy, where one or more chromosomes are in extra or less in number. And the last one is polyploidy, where there will be an extra of complete set of chromosomes. Now we will go into details of each of these types one by one. So let's go. So let's start our discussion with rearrangement. At the name itself suggests, it rearranges the segments within a chromosome. So the very first is duplication. So again I will be taking this example, ABC centromere DEFGHI. So duplication means a particular segment of the chromosome can simply be duplicated. So let's say ABC dot D DE, and this DEF segment gets duplicated. DEF, and then GHI. So now you can see, sorry, let me write that in the original black colors. So now you can see that this particular is the duplicated segment, right? So the duplication can be tandem, meaning it can be next to each other, right? Like in this example, or it can be displaced. If it were a displaced duplication, it would be somewhere far off or away from DEF. So let's say DEF GH followed by DEF and then I. So that would be the case of displaced duplication. Now there are two repercussions whenever such rearrangements happen. The very first thing is the gene dosage. Now you know that genes or DNA goes to RNA, goes to protein, right? So if the number of DNA is more would be the number of proteins, and proteins are involved in a lot of reactions, and hence the chemical reactions in which the proteins are involved, the reactions would be disturbed. This is the very first side effect of duplications or any other chromosome abnormality, is the gene dosage that would be affected. Another thing is during meiosis. Now we know that the chromosomes need to pair, or even in case of mitosis, the chromosomes need to pair during mitosis or meiosis. So whatever we want to talk about, mitosis or meiosis, the pairing would be affected by any of these changes. Now I want to talk about the next one, that is deletion. Now it's not very different. All you have to do is delete a segment. So let's say I'm writing my original ABC dot DEF is no more, GHI. Now deletions can be pretty harmful, right? So if you have a gene that was very important, that a deletion could entirely destroy the protein, right? Because there is no more protein. Sometimes a segment of protein would be lost, or sometimes even a complete protein can be lost, right? And it can lead to non-functional proteins. Inversion, as the name itself suggests, a segment of the protein just goes opposite. So DEF can be written as FED, and the remaining segment is same. So as you can see, the inversion can happen para, that means next to. Para means next to centromere, as in case of this one. It is next to centromere. The centromere is not involved. Or it could be A, B, E, F, C, D, G, H, I. So here a centromere is involved. So this is peri, around the centromere. So that's the another kind of inversion. The next one is translocation. Again, a very easier translocation. That means a segment of your chromosome would be translocated, shifted to another chromosome. Now again, it could be just shift from one to another, or I can say non-reciprocal. So only one chromosome loses a segment and the another gains, or it could be reciprocal. That means they exchange these segments with each other, right? So this is the first type of chromosomal variations that we talk about. Now let us talk about the second type, that is aneuploidy. Okay, so aneuploidy is pretty simple. What it means is you have more or less number of chromosomes. So nullisomy means one complete set of homologous chromosomes. If you don't remember, I would like to remind you, homologous means the chromosomes that you get from mom and dad and those which influence the same characters, right? So we have 23 sets of chromosomes, or we can say we can have 23 sets of homologous chromosomes. So nullisomy means, let's say 44 chromosomes only, right? So you only have 44 chromosomes. The complete 2 chromosomes are lost. Monosomy is when one chromosomes are lost. So let's say 44 plus X, and because we don't have the 46 chromosomes, it is represented as O. This is a case of Turner syndrome. So monosomy means you lose only one chromosome. Trisomy means you have an extra. So let's say Klinefelter syndrome is an example of trisomy. So 44 plus XXY. So you see that we have an extra X chromosome here. Tetrasomy, again the name is very clearly answering the thing that you have a complete, an extra set of homologous chromosomes. Now, because you have more or less chromosomes, so if you lose a chromosome altogether, then certain characteristics would be lost. Most of the times when the autosomes are lost, so because you have two types of chromosomes, autosomes which do not determine your sex, and then you have sex chromosomes. So autosomal aneuploids are less bearable but more frequent in humans. Okay, let us talk about the last one, that is polyploidy. Now the word itself says poly, that means more than two sets. Now normally we have two sets of chromosome, as we just discussed, that is one from mom and one from dad, right? Now polyploidy simply means that you have more than two sets of chromosomes. Now autopolyploidy, that means you have more than two sets but of the same individual. right? And if the source is different, then that's called allopolyploidy. Now, certain times we want to introduce allopolyploidies because we want characters of two different species, or and we have tried doing this with Brassica oleracea. We have tried this, and Raffinus sativa, that is your radish. So we have tried doing this, but again this was not successful. Actually, we know that brassica, that is the cabbage, we only want the leaves from the cabbage, and radish, that is Raffinus sativa, we only want the roots. So that is why a scientist wanted to get the best of both, but unfortunately the opposite happened and it did not work. So autopolyploids and allopolyploids are such, but there's a key thing that you must understand. If we have even versus odd, so if you have odd number of sets, this doesn't help us because the odd brings the problems during meiosis. It will be problematic to reproduce, right? If sexual reproduction has to do. But again, we have successfully done this in case of bananas. Now, like bananas, we want seedless bananas, right? So we had a variety that was an odd allopolyploid, and that's good because we don't want seeds, we just want the fruit, right? So that works. Even polyploids have better chances of reproducing by self, but again, we just look for the commercial features. To summarize all of this, so basically in today's video, we talked about the three types of chromosomal mutation, that is rearrangement, aneuploidy, and polyploidy. We talked that rearrangement basically talked about duplicating a segment of the chromosome, deleting them, inversing them, or simply shifting the location from one to another, both at the same time, reciprocally or not. In the aneuploidy that we talked about we might have more or less copies of a one or more chromosomes, whereas when we talk about polyploidy, we talk about more or less sets of chromosomes. I hope this makes sense. Thank you.
Hope you guys like this video. Make your geek happy. Subscribe to our channel.